right. Well, good morning, good afternoon, good evening, everyone, and welcome to today's World's Most Exciting Classroom event. My name is Joe Grabowski, and I'll be your host for today. Today's Darwin 200 event is a little bit different than usual. I'm coming to you from a mountain wilderness hostel in Alberta. We are in the Rocky Mountains, so I'm out with a team of explorers, and we will be looking at freshwater, exploring the mountains, so it's going to be a really fun few days. So it's a pre-record for today. I've gathered some great footage from the ship, from some Darwin leaders in Fiji, some updates from Grant in the field. We've got our experiment, our curiosities. So everything we usually have is coming your way, just in pre-recorded fashion. So let's do a really quick catch-up on where the Oosterschelde is right now. I'm going to share my screen. There we go. So you can see that we are about halfway between New Zealand and Fiji right now. If I get in a little bit closer, we can take a little bit of a better look. There we go. And then let's zoom out a little bit. So there's Fiji. You can see New Caledonia up there. Australia starting to appear there. And if we back up just a little bit more, you can see we're starting to get close to the North Island of New Zealand. So there is where the Oosterschelde is right now. Our Explorers Club researcher on board for the leg from Fiji to New Zealand is Eric Herrea. So Eric is looking at eDNA, or environmental DNA. So he's taking samples on land in the soil as well as in the water, and he can tell what organisms have moved through that soil or moved through the water. So we have a little video clip from the ship of Eric sharing what it's like to do this eDNA work. Let's take a look at that clip right now.
Hi, I'm Eric Herrea, and I'm from Mexico, and I'm here on the Oosterschelde as part of the Explorers Club, and my project is collecting environmental DNA and environmental RNA. And what's really cool about this specific project is that the kit for collecting DNA will actually stay on the ship until it reaches port back in Falmouth in England. So we'll be able to collect DNA and RNA samples and essentially be able to map all the genetic diversity along the path of the ship, which we're super excited about. And while it sounds really interesting, really fun, the actual collection is as simple as can be. And I'm going to do a collection right now. So we're currently in transit from Fiji to the North Island of New Zealand, and we're collecting water samples. So we have two samples that we can collect. Samples as either liquid samples from water or from solids, and today is a liquid sample. So this is actually how you do it. So DNA is actually negatively charged. Means it's attracted to positively charged things. In this case, this is what we collected on. It's a positively charged membrane, and we pass water through it until it clogs up with DNA. And the little liquid you see inside here is a very high salt solution, and that high salt allows us to keep the DNA stable for long periods of time. And so we just brought up this bucket of water from the side of the ship, and we use sterile technique, or as sterile as you can get on a ship. Pull up water, pull up the filter, and then force the water through the filter. And this is the fun part. You just do it until the filter is essentially clogged, and it has very, very small holes, 22 billionths of a meter, and that allows very few things to get through, including DNA. So DNA is stuck there until we elute it, and eluting just means that we use another chemical to solubilize it and actually put it back into solution. And we'll do that back in the lab. And it gets harder every time you push. As the DNA actually clogs, you can actually stop the water from going through. I think we'll be able to do about 2.5 here. And so everything on Earth, every living thing has DNA and RNA. And what those do are essentially the instructions for life. So you and I have the same DNA, but sometimes it gets expressed differently, and that's what makes proteins. That's what differentiates hair from skin cells, from liver cells. However, in nature, you have diversity. You have diversity in hair cells, for example. every single mammal will produce a different type of RNA for a different type of hair cell. And that's what we hope to collect. So we're putting it back here in the preservation solution, and this will stabilize it for about 5 years. We are not gonna wait that long. We're actually gonna take it back to the lab in Texas and actually be able to process it fully. But part of DNA is everything is found in a context. So in this case, what is the pH of the solution? We measure it with pH indicator paper. And, oh, splash. Part of being outside the lab, we get splashed. So we dip it in and we can actually see what color change does it get. It's pretty fast. In this case, we're seeing color is around 6. Good to know. And temperature. So we hit it with our infrared thermometer. And we're seeing a pH of 6 and a temperature of 19.9. So, as the Mythbuster said, what makes science science is writing it down. So this is sample 7. Water, pH 6, temperature 19.9, and then we go up to the captain and ask what our coordinates are, and that is it for the sample. Now what we would do is, back in the lab, we solubilize the sample, where we can actually extract the DNA, separate out the DNA from the RNA, and the RNA then gets sequenced. So we run it through very, very small proteins and actually shock it. And every time it shocks it, we get to hear a different sound, essentially, a different wavelength. And what that tells us is A, T, G, or C. And those are the 4 letters that make up DNA and RNA. RNA is actually a U, but way too complicated. So essentially, that's what tells us the genetic sequence. Once we know the genetic sequence, we can then compare it against known sequences. And we can get one of 3 options. It's either a known sequence, for example, a muscle cell of a tuna fish. That's been identified. The other one is essentially, it's a muscle cell of a fish that has not yet been identified. And it's a variant. And the third, which is my favorite, is unidentified non-variant. And essentially that tells us it is completely new to nature. And given diversity, we expect to find a lot of each of the 3. And the last one is the most important one because it actually gives us the diversity to test new machine learning models. Whoever controls the most data can train the best machine learning models, and those machine learning models for biology are insanely cool. So you've heard of ChatGPT, where you can say, or DALL-E, for example, make me an image of an apple, or a beaver eating an apple on a bicycle, and it'll do that. What if you could do the same thing with life, and you can tell it, make me an enzyme that degrades plastic, or make me an enzyme that solubilizes certain metals so that we can collect heavy metals from water. That needs a lot of training data and a lot of diversity, and that's what we're doing here. We're training data, we're getting training data for these models by actually sampling life, 1 bucket at a time. Thanks.
So we have an update here from Grant. Grant is in the field in Fiji. He's looking for bird species on land, looking for endemic species found nowhere else in the world other than on the island in Fiji that he's visiting. So he sent us a little update from the field. Let's check it out now.
Hi everybody, Grant here from DARWIN200, and I'm reporting from the middle of the Taveuni rainforest. It's definitely been pretty rainy in the rainforest today. I've been filming birds here for my upcoming documentary project, Pacific Birds Evolution, Extinction, Survival. And I'm really excited to tell you that I've seen a lot of the cool species that I've been after, and I've gotten some cool shots of some of them as well. Hopefully I'll get a few more by the end of the day. It's a beautiful island. They call Taveuni the Garden Island. It's a really wet island. It traps a lot of the moist ocean air that flows this way from the south. And as a result, you get these lush forests that are home to quite a lot of really interesting birds. And hopefully over the next couple of weeks, we'll get to share some of this with you. Take care, everybody, and hope everyone is doing well back home.
So it looks like Grant's having a good time in Fiji, and we're going to stick with that theme right now. Last week, I caught up with the two Darwin leaders who joined us in Fiji, working on their projects in the field. The first one we're going to check out is Alexia, and a little bit later, we'll meet Vinny. So for now, let's check out Alexia's project. All right, everyone. Well, we are live with the Oosterschelde. We are in Fiji, and we have one of our Darwin leaders on board. We've got Alexia from New Zealand. Alexia, how you doing?
Full of Vanaka, which is hello here in Fiji, and hi everyone. I am so excited to be here with you all and on the Darwin Oosterschelde, as Joe so clearly said. Better pronunciation than me.
All right, perfect. Well, look, we want to learn all about you. We want to learn about the work you've been doing this week. So I think the best place to start is if you just want to introduce yourself, tell us a little bit about what you do when you're not hanging out in Fiji.
Yes. Hi, everyone. My name is Alexia. I am part Samoan, but I live in New Zealand. And if you've heard of New Zealand, let me know in the comments. But I am a storyteller and conservationist, and also the founder of an organization called Girl Boss New Zealand, which is all about encouraging young women to step into careers in science, technology, engineering, and maths.
All right, very cool. Well, we love to hear about how people found the Darwin 200. So how did you learn about the Darwin 200 and what made you decide to apply to be a leader?
Yeah, absolutely. So I was so lucky to hear about the Darwin 200 through National Geographic, as I'm a National Geographic Young Explorer. And I am so passionate about using science as a way and storytelling as a way in which we can get lots of young people like yourself excited about a career in science. And so when I heard about this opportunity to create a film and raise awareness about a very, very special animal, and so I'll tell you a little bit more about my project when I'm here in Fiji. So this right here is a Fijian $100 note. Okay, this might look a little bit different to the $100 note in your country, but here it is, here in Fiji. And if we turn it to the other side, we can see it actually has this little bug right here. Can you see that? And anyone wants to guess, let me know what you think this little bug is. Some people might think it's a cockroach. It's not a cockroach. It's not a beetle. It is actually a cicada. So you may have cicadas where you're from, but this is a very special cicada. This is the Fijian cicada. And what they call this Fijian cicada here in Fiji is the nanai. And so I'm here to raise awareness and help people get excited about this little bug. And this bug is very special because it spends 8 years underground, and then only after 8 years it comes up and it emerges from the soil and spends a few weeks flying around, and then after a few weeks it dies. So this is a very special bug for spending 8 years chilling underground and then a few weeks up above. And why this is such a special insect here in Fiji is that it's only native to Fiji. So the only place in the whole world you can find this very special cicada is here right now in Fiji. And so we are raising awareness because next year, 2025, is the year in which the nanai rises from the ground. So you have to wait a whole 8 years to see this special bug. And what happens is it rises from the ground, and then people go out, and you know what they do? They pick it and they eat it. So I don't know if you like to eat bugs, but here in Fiji, they will eat the cicada, and they say it tastes like shrimp. So I'm not sure if that's something you want to do, start eating cicadas, but that is what they do here in Fiji. And what's so special is that they say that the year that the nanai rises, 2025, is a year of good luck. So next year, it's a year where all your dreams can come true. And this is actually based on some history which is being passed down from generation to generation to generation here in Fiji. And where this came from is that because the cicada would rise from the ground in the year of the Nanai and fly around and then die, it would die in the water. And it would die in the water, and then the fish would come and they would eat the cicada. And once they eat that cicada, then they get really fat. And they eat so many cicadas that they get really fat. And so then all the fish are fat. So that's why they think, wow, the year of the Nanai is a year of abundance. It's a year of fat fish. Because when the fish eat so many cicadas, they get too lazy and they've eaten too much like how you might feel full after Thanksgiving or Christmas Day, and therefore they're easy to catch. So this is the story of the Nanai.
All right, Alexia, that's really cool. You're going to have to go back in a year and you're going to have to try them for us and let us know if they taste like shrimp or not. So.
I know. I don't normally eat bugs, but this is a pretty special bug, so maybe I'll have to make an exception.
All right. Well, you were in the field. You were learning about the Nanai. You were talking to local conservationists. So, what was your week like? What was it like in the field?
Yeah, absolutely. What's so special about doing this work is that I got to travel right up into the hills, and it was quite the trek. And there was a lot of holes in the ground, so there was a lot of bumping as we're going up the hills. But I actually got to meet these elders that live in the hills of Fiji, and you'll be able to see that when you watch my video that I created about this in just a moment. And it was amazing to be able to sit with the elders and to learn from them, because as we know, some of the most special advice can come from the older people in our lives. For example, our grandparents, maybe our parents, and they can share some wisdom that we can pass down. And so to sit with them and to hear their stories and to hear about how they have lived through multiple Nanai years, it was really, really inspiring.
All right. So you set the stage pretty nicely for us talking about your video. So I say we take a look at it and then we'll come back and we'll have a few more questions together. So I've got it here on my end. All right. So here we go. Here it comes.
I bet you know what this is. It's a Fijian $100 bill. But who's this little guy?
A cockroach? A beetle? No, a nanai.
This is the Nanai. It's on the Fijian $100 note because this little bug is kind of a big deal. The Nanai, or the Fijian cicada, spends most of its life underground. Once every 8 years, something magical happens. The Nanai rises from the earth, sheds its shell, and reveals delicate wings. For just a few short weeks, it sings a song of love, calling out for a mate. So why is the Nanai on the Fijian $100 bill? To find out, let's learn the legend of the Nanai. Born from the Boronito and Noimalu clans in the highlands of Viti-Levu in Fiji, the legend states that the year the Nanai emerges is a year of peace and abundance. 2025 is the year of the Nanai. It's re-emerging after 8 years. It represents rebirth and is a unique opportunity for you to shed the old and step into the new. Because 2025 is the year you make it happen. So we want to know, what's your dream for the year of the Nanai?
I want to become a certified plumber.
I want to become a professional rugby player.
Whatever your dream is, we want to see you bring it to life. Post it and use the hashtag Year of the Nanai.
All right. Alexia, that is a pretty cool video. I can't wait for the rest of them now.
Yes, it is. And it was so fun making that video, especially doing all those costume changes, as you may have seen.
Very, very cool. So you were out, you were talking to people in the community. I wonder if you can tell us a little bit about conservation. Is there threats that I know they only come out of ground, I think you said every 8 years. So is there threats that they're facing while they're kind of underground and waiting to hatch and emerge?
Yeah, absolutely. So we were looking at this from a conservation lens. And one thing that made it a little bit difficult is that because the Nanai spend so many years underground, we don't exactly know what's going on with it. We don't know how many are under the ground exactly, and we don't know really how they're doing. And because it's only every 8 years, what we want to do is we want to make sure that we track next year are they how they are, how many there are, where they're coming from. And so that's why we're already going around and hearing stories from people who are farming in these communities to say, have you spotted any of the little nanais before they emerge? And we have heard stories that already they are in the ground about 30 centimeters right now. But by the time of 2025, there will be thousands of them emerging. So what some of the risks that can impact the Nanai is obviously deforestation. So we need to make sure that we are advocating for the Nanai and getting people excited about it so that we can reduce these risks, because we do not want to take away the Nanai's home.
All right. So you're making your 3 videos. The first one's ready. It looks amazing. I think the other 2 are going to be pretty fun as well. But I'm curious to know a little bit about what it was like making the video, or maybe talking to the community members. What was the most challenging thing about your experience as a Darwin 200 leader?
Yeah, I think the most challenging thing about being a Darwin 200 leader, I think the most challenging thing was I had so many ideas for what I wanted to communicate in my video, but I also wanted to make it short, because I know that with things like TikTok, some of you probably a bit too young for TikTok, but I'm sure you know TikTok, or Instagram Reels, we're so used to watching short videos. And I wanted this video to reach young people like you, and to make it short and make it fun and make it high energy so that you can learn more about the Nanai. And so that was a challenge, because I had so many ideas, but I couldn't say everything I wanted to say. I had to get it really tight and really shrunk down into just 90 seconds to get people excited about the Nanai.
Okay. And so your experience is wrapping up. Time always goes so quickly. And you'll be back in New Zealand. So I'm wondering, Alexia, when you're back, what's next for you? Do you have something on the horizon you're really excited about, an event, a project? What's going on in your life?
Yeah, absolutely. I'm leaving Fiji soon, but I'm excited because I can see my family back home in New Zealand, because I've missed them so much. And when I get back, I am back into my job, which is at Girl Boss New Zealand. So we are running a big event for young women to get them excited about science. So if there's any girls listening to this, I want you to know that we want to see more of you step into science and realize just how awesome you can be in that field, because as we know, girls can truly do anything. And also, I am excited to stay connected to the charity that I'm working with here in Fiji, which is Nature Fiji. And this charity are doing amazing work. And it is led by this incredible boss woman who, Nunia, and she is an incredible woman. And she started Nature Fiji because she saw a problem and she decided to step up and solve it. And so I want to challenge all of you listening now, if you see a problem in your community, then you actually can be the one to step up and solve it. You don't need to wait for someone else to do it. You can actually be the person who makes the difference.
All right. I love that message, Alexia. I'm so glad you had a great time in Fiji. Sounds like you learned a lot. You made some new friends. I mean, what could be better? We're looking forward to the rest of the videos. And a huge shout out. Thank you so much for taking some time to hang out with us and share your experience.
Thank you. I had so much fun. And I want to teach you one last word before I go. And that is the Fijian word for thank you. So the Fijian word for thank you is vinaka. So Joe, can you say vinaka?
Vinaka.
Yes, vinaka. Yeah. Thank you, everybody. Have a great day.
Thanks, Alexia. Okay, so 2 weeks ago we had our experiment polishing your own stones using a rock tumbler. So let's take a look at Stu's results from the Darwin studio, and then we've got a brand new experiment coming your way right afterwards.
Welcome back, everyone. Well, my efforts to create beautiful polished stones are going well. My tumbler has been tumbling for nearly 2 weeks. As you might recall, when I set the experiment up, I started off using a thick coarse grit, and I set it going using that for about 6 days. Then I used progressively finer grits to give my stones a more beautiful polished finish. 3 days ago, I set it going with polishing foam, and that will hopefully give the stones a really gorgeous, glossy finish. Well, it's down to 1 day. I think it's time to end this. So let's have a look at how the stones have ended up. Let's open up the drum of the tumbler and remove this plate. So the first thing you need to do is sieve out the stones and separate them from the water and the polishing foam. So let's have a look. Hopefully, yep, there are my, wow, they look beautiful. So one by one, let's put them on this paper towel and give them a good dry. These really have turned out beautifully. Let's put the sieve to one side. And look at our beautiful stones. As you might remember, I put in a mixture of different types of stones. I put in several types of tiger's eye. This is a beautiful mineral with this gorgeous, glossy, iridescent shine. I put in some standard orangey brown tiger's eye, some of this gorgeous greenish tiger's eye, and even this reddish tiger's eye, and this blue tiger's eye. You can see that iridescence really well. And that polishing foam has given them this gorgeous glossy finish. That's pretty cool. I put in some of this blue agate. This has come up really well too. It really shows off the colours. I also put in a chunk of labradorite. This is famous for this iridescent shine. And you can see now with this polished finish, that iridescence is really clear to see. And look, that looks gorgeous. I think that's my favourite so far. I also put in some regular stones. And okay, these aren't quite as shiny as the rest, but they still come up pretty well. The effect of the tumbler is a bit like the waves of the sea, washing up stones on a beach, causing them to have this beautiful rounded shape and polished finish. You can even turn your beautiful polished stones into jewellery. You can easily buy these beautiful necklace casings and place one of your polished stones inside of them. Or you can affix them to rings such as this one, or glue them to earrings such as these, and make your very own gemstone earrings. Well, I think my polished stones turned out pretty well. I'm quite proud of these. How did you get on? Were you able to similarly make beautiful stones such as these? Send in your photos to classroom@darwin200.com. I'd love to see how you got on. Thank you for taking part in this polished stones experiment.
Okay, those stones turned out pretty well. That's pretty cool. It was great to see some photos come in from some classrooms. A few classrooms were able to do it, as well as some students showed off their polished rocks that they had at home, that they found in museums and other places. We have a brand new experiment coming your way. 2 weeks to give this experiment a try, and send us pictures to classroom@darwin200.com. Let's take a look at the brand new experiment.
In today's experiment, we're going to create a wormery, which will allow you to study some of Charles Darwin's most interesting research. In the last few years of his life, Charles Darwin wrote this book, studying the impact of earthworms on the production of soil. It's one of the least appreciated, but arguably most important works of the life of this great naturalist. To create your wormery, you need a container, some soil, some leaves, and some worms. I bought these worms from a fishing shop. These are actually very lucky worms because they were going to end up on a fishing hook as bait for fish. But instead, they're going to end up in my wormery and eventually I'll release them. So they don't know how lucky they are. Although as you can see, they're trying to escape. Before you put your worms into your wormery, you can have a good look at how they move. It's pretty amazing how they use rhythmic movements along their body to propel themselves forward. These guys are surprisingly quick. Look, they're trying to escape. I'll put you back in there. To make your wormery, you need a see-through container, such as a jam jar or plastic bottle or container, such as this one. First, add in a good amount of soil. Fill it about 2/3 of the way. Let's put in that very carefully. Any old garden soil or potting compost will work fine. Make sure it's nice and moist for your worms so they're happy. Let's spread that out evenly in there. Then you can add your worms. I put these in a paper plate so it's quite easy to pour them. Here we go, worms, in you jump. There we go. The last few, come on you. You don't want to go in the wormery. I think you'd be happier in there than on a fishing line. There we go, last 2 worms, in they go. And then, last but not least, put in some dead leaves. This leaf litter is very important. You'll see the action that the worms have upon it. You'll notice that the soil and the leaf litter form 2 very distinctive layers because of the way that we've put it in. You can then either put on the lid or, to be honest, what's better is a piece of plastic with an elastic band. You can then punch a few holes in the top of the plastic. This will allow air exchange, and that's really important for the worms so they can breathe and live healthily. You then need to find somewhere to place your wormery. Make sure it's not in a place that would be exposed to extremes of temperature or really strong sunlight, because that can cause the wormery to overheat, and the poor little worms won't like that very much. A shady windowsill could work really well. Over the course of 2 weeks, observe the wormery really carefully. Do the worms move around, or do they stay in one place? And what happens to the leaf litter? I can already see many of my worms are really exploring their new home. Join me in 2 weeks' time and see what my worms have got up to. I hope you enjoy making your own wormery and observing your worms. Good luck and thank you for taking part.
All right, we are going to head back to Fiji and we're going to meet Vinny from Tasmania. She was our second Darwin leader. She was on board the ship and we had a little interview together. Let's check out her project with the Fiji sharks.
Hey everyone, we are live here in Fiji on the Oosterschelde, and we are catching up with some of our Darwin leaders. And I'm really excited to have Vinny here live with us. Hey Vinny, how are you?
Good, thank you.
All right, well, it's so great to have you live with us, fresh off of your work in the field as a Darwin leader. I think the best place to start is, take a minute, introduce yourself. Tell us a bit about you.
Yeah, hi, my name is Vinny. I'm from Australia, and Sorry. Hi, I'm from Australia. My name's Vinny. I'm from Australia, and there I'm part of an organisation called Jane Goodall Institute Australia. I'm the state coordinator there, and I help. And the whole philosophy of Jane Goodall Institute is that we help connect people with the environment and animals, and that they all need to work together in cohesion in order to have a balanced and sustainable world going forward. I studied microbiology at university and I grew up in Tasmania, surrounded by water, which means that I'm a water baby, love snorkeling, which was really great because my project for the DARWIN200 was around sharks. So it sort of matched up a bit.
We're always curious, how did you learn about DARWIN200 and what inspired you to apply?
So I heard about the DARWIN200 program through the Jane Goodall Institute actually. So our coordinators manager came up to me, he's like, hey, I know you love the water, you're so passionate about it. There's this great opportunity where he sold it like I was going to be on the ship sailing around the world. And I was like, oh, that sounds interesting. Read it up and I was like, you're not quite sailing around the world, but it's still really great. So that's sort of how I heard about it. And the reason I wanted to apply was I've always loved nature. I've always loved being out and about, hiking, snorkeling. I suppose it's a side effect of growing up on an island. In Tasmania, we have some of the most incredible waterfalls, the most incredible waters to swim in, but also some of the most incredible hikes that you'll ever find. I might be slightly biased, but I think that's true. So,
Vinny, I'm 100% with you. I visited Tasmania. It is an absolutely beautiful island. I had a ton of fun there, so I can see why you love it so much. It is the nature, the wildlife. I saw Tasmanian devils in the wild. It was pretty fun. So I wanted to hear about your project. Vinny, tell me a little bit about the group you worked with and what your week looked like.
So my project was looking at the Fijian sharks, and that's about all I was given. From there, it was my creative liberty to go and figure out what I wanted to do, which angle I wanted to take. And I actually looked at sharks and their cultural significance to the Fijian community. So sharks actually hold a really strong connection to Fijian culture and tradition. They're often the totem animal for many communities, and they're seen as the protectors of the sea and the protectors of fishermen. They guide fishermen back safely in a storm or when things go bad. In some Fijian communities, there's a Fijian god who would shapeshift into a shark and would protect and look after the Fijian communities. So sharks hold a really strong cultural significance in this area. And while out in the field, I also came across this concept that the waters, the Fijian waters, they belong to the people. So any sort of protection has the community at its forefront. So basically, everything is really community led, and I really appreciate. I loved that. That was one of my biggest highlights when I started doing my field work. And so taking that, I decided to do my project on how shark conservation needs to have community in its center, and how we can have the community at the forefront of its protection strategies and how we protect sharks.
All right. Well, speaking of your project, let's take a look at the first video together now, and then we'll have another question or two before we wrap up. So here we go.
Fiji, a tropical oasis of beautiful beaches and lush forests, rooted in culture and tradition. But it's not just a haven for travellers. It's also the home to the world's most misunderstood creature, the shark. Wild and wonderful, sharks are the heart of their ecosystem. Seen in lore and legend as the protectors of Fiji, they are integral to both Fijian culture and their marine ecosystems. But despite having no natural predators, sharks are under threat. I'm Vinita Gupta, conservationist, storyteller, and Darwin leader, and right now I'm in Fiji, diving into the world of sharks. These wild and wonderful creatures have roamed the oceans since long before the time of the dinosaurs. Sharks play a vital role in the health of the ecosystem. By preying on herbivorous fish, they prevent overgrazing, allowing seagrass and coral to grow and flourish, maintaining the balance of the whole ecosystem. As well as being a keystone species in the habitat, sharks have a unique and special place in Fijian culture and tradition. They are the protectors of fishermen and the local communities.
My mom is from the second mainland called Vanuatu. So this legend, and also they always tell us stories about the shark out of Fiji in the Kuanga. And since I was a kid, I started believing it. But once I get older, it's not in me. So they always tell me that they are our protector when we go out, especially for them because they are fishermen. So they believe that when they go out, the sharks protect them from very dangerous animals underwater, or if they just stop out there, they're always there to take them back to the mainland.
Although sharks hold a special place in the Fijian culture and their own unique ecosystem, not all is okay. These apex predators are under threat. Fishing practices such as gill netting, overfishing of shark prey, pollution, ocean acidification, and climate change pose significant threats to this incredible creature. On top of this, it is hard to properly evaluate the threat severity due to the lack of reliable long-term data. Historically, shark research in the area has been limited and far between. One of the many reasons for this is the confusion of who's responsible for the monitoring and protection of Fijian waters. In Fijian culture, the coast is owned by the people. Local communities are entrusted with the protection and guardianship of the coastal areas. However, communities don't have any legal rights over the coastal areas, meaning that without support from the government, the local Fijian people don't have the means to enforce protection of the waters. Data on the distribution of Fijian shark populations and their behaviour is limited and has gaps, which makes determining how severe the threats to sharks are difficult to fully understand. But not all hope is lost, and there are people working hard to change that. This film series will look to fully understand and find solutions to the problem of how to bridge the gaps in the current shark data. In the next episode, we will delve further into the complex problem and meet some of the people dedicating their lives to protect these amazing animals.
So Vinny, you spent the time in the field. It looks like you had an incredible time. You met some amazing people. What was the biggest challenge you faced to try and put this video together with your work out in the field?
So the biggest challenge, other than the heat in Fiji, because I come from a very cold and temperate climate, so having to deal with the tropical Fijian heat was a challenge in itself. But other than that, I think the biggest challenge I faced was, as part of my project, I was working with an incredible organization, IEC and Oceania, and I was looking at a really incredible tool that they're developing called the Marine Spatial Planning Tool. And it's an incredible tool, but it's also a very complicated tool. And with the format of how the Darwin program is made and how we communicate our findings in film, it was quite a challenge to take this tool and actually create something that's easy to understand, isn't going to take 20 minutes to explain on film, and communicates how important it is for the Pacifica, because this tool is uniquely made with the Pacifica community in mind, with their unique sense of how they work together to protect the oceans. So like I was saying before, the Pacifica community and the Fijian community as well specifically, they protect their waters. They own their waters. And so this tool really encompasses that, and trying to take that idea and communicate it in a way that's easy to understand, but also really encompasses the essence of that whole philosophy, was probably one of the most difficult things I found in the project.
All right. And so you've had an amazing time as a Darwin leader. I'm so jealous. Fiji's such a beautiful spot. But now you're going to head back to Tassie, back to Tasmania, back to Australia. So, what's next for you? Do you have a project you're excited about? An expedition? What are you up to next, Vinny?
I think I'm really excited to go back and be able to actually share my experience and share my findings. So I don't think many people really understand how integral the coastal areas are to the Pacifica community, and being able to take those learnings back, and actually being able to tell other people about them. I'm really excited to be able to do that and actually say, hey, there's some really cool traditions and culture, and actually be able to spread that out, because I think it's so impressive how over so many generations, so many years, the Fijian community have managed to not only keep a stronghold on their values, but also pass them on.
Well, Vinny, it sounds like you had an incredible experience. We can't wait to see the rest of your videos and learn more about the sharks of Fiji. Thank you so much for letting us steal a little bit of your time while you visit the boat today. All right. Thanks, Vinny.
Thanks. Bye.
We have a brand new curiosity of the week to check out, but before we do, we have to take a look at last week's curiosity. So last week's curiosity was an abalone shell. They are a marine species of sea snail, a gastropod mollusk. They have these beautiful iridescent inside their shells, which makes them highly prized in many cultures. Unfortunately, due to their great taste as well as their prized shells, they've become endangered species in many parts of the world as the demand for them has increased. So in many places, wild abalone fishing has been banned, or there's quotas in other areas. And of course, there's some areas of abalone farms where they're trying to sustainably harvest and raise them. So we have a brand new curiosity for this week to wrap things up. Let's take a look at it. Remember, you have 1 week to send in your answer to classroom@darwin200.com. Let's take a look at this week's curiosity from Stu in the Darwin studio.
This week's curiosity are these objects here. Can you guess what on earth these strange but intricately patterned objects might be? If you look very carefully, you'll see there's a couple of prominent holes, as well as a very intricate pattern all over this strange object. One other clue, it is a type of animal. But what animal do you think this could be from? It's definitely not a skull. Send in your best guess to classroom@darwin200.com for a chance to win a £50 Amazon gift voucher prize. Join me in 1 week's time and we'll find out the identity of this very strange pair of objects. Good luck and thank you for taking part.
All right. Well, thanks everyone for joining us for this week's World's Most Exciting Classroom event. Next week we'll be back to normal. We'll be full on live, but I hope you enjoyed our pre-recorded content this week. We had a lot going on, from our leaders in Fiji, catching up with Grant, checking out the sharks. What else did we do? We had our new experiment, our curiosity of the week, Eric on board doing eDNA. There's never a dull moment with the Darwin 200 crew. Thanks everyone for tuning in. The last thing we need to do is a big shout out to our sponsors, our supporters, our partners, without whom we wouldn't be sailing around the world. We wouldn't have research going on the ship, the world's most exciting classroom, or bringing Darwin leaders out into the field to train the next generation of future conservationists. So we'll wrap up for the week with a shout out to our partners.